What's up, nerdlings? Today I'm going to be talking a little bit about meiosis. So, how do organisms reproduce? Well, they employ two different strategies. Organisms can either reproduce sexually or they can reproduce asexually. So, as far as asexual reproduction goes, A in biology means without. So, asexual reproduction means without sex. So this can happen in a couple of different ways. Some organisms employ the use of a process called binary fission. This is very similar to meiosis, and it results in just the same type of offspring as the parent. So kind of like mitosis, it's going to result in identical offspring or daughter cells. So one of the examples is going to be this little guy right here, an amoeba, and he's going to undergo a mitosis-like process called binary fission, which results in two identical daughter cells. Now, we also have a process called budding, which is basically when an organism buds off into a new organism, such as you see in this sea anemone. So obviously, there's pros and cons of asexual reproduction. One of the pros is that it does occur pretty rapidly, so those organisms are able to reproduce at a very rapid pace. Those offspring are typically adapted for the environment in which they come into existence, and they don't have to find a suitable mate and expend energy in doing so. Now, some of the cons is that the fast reproduction can actually lead to overpopulation of that organism. There's not going to be any genetic diversity, which means if there's some type of disease or natural disaster that affects that population, you're not going to have one of those organisms being more adapted to the environment, meaning that they would live to survive. All of those organisms are going to get wiped out if they're all susceptible to a specific disease or some type of environmental condition that occurs. So in sexual reproduction, parental organisms produce haploid gametes. Now, when I think of the word haploid with my students, I always say haploid means half, meaning that that organism or that gamete only has half the number of chromosomes that the parent itself does. So gametes form, and gametes will fertilize. So if you look right here, we can have a sperm cell as well as an egg cell. Those are called gametes. So specifically, sperm cells are called spermatocytes and egg cells are called oocytes. So the developing organisms go through differentiation to get different cell types, meaning that once that egg has been fertilized by a sperm, we go from haploid, so having half the number of chromosomes, meaning sperm and egg, they only have half, they're haploid. When they come together, they form a whole, and we call that a diploid organism, meaning that it has two. So in biology, di means dos, or two. So it's going to form a 2N zygote. And then that zygote can divide, and again, it can differentiate, meaning that some of those cells are going to become liver cells, and some cells will become blood cells, some cells will become skin cells, eye cells, brain cells, all kinds of cells. So again, just like with asexual reproduction, there's going to be pros and cons. So some of the pros are that this is going to lead to genetic diversity, meaning that if there's some horrible disease that goes through the population or a natural disaster, there will be some of those organisms who are different, and they might be better adapted to the environment and survive and be able to pass on their genes to future generations of offspring. So it does also prevent against quick overpopulation. Most of the time, sexual reproduction is a lot longer of a process. So if you think about it, humans, our gestation period is approximately 9 months. So from start to finish, it takes approximately 9 months in order for that fertilization to occur, and then the zygote to mature into a fetus, and for that fetus to be born as a cute little baby. Although sometimes So some of the cons are that obviously, yes, it does take longer, but the offspring isn't necessarily adapted for the environment. Meaning, unlike asexual reproduction where it's an identical copy, and if that organism is better at surviving harsh changes, so for example, tidal populations that might be adapted for fluctuations of the water levels. in sexual reproduction, that organism isn't necessarily an exact copy of the parent, so it might not be as well adapted to the environment it's placed in. And also, finding a suitable mate, as many of us know, is a very difficult and time-consuming, as well as energetically consuming process. So going over some meiosis terminology, like I said in the previous slide, we have haploid and diploid. So haploid means half, or half the number of chromosomes. In a diploid organism, you have two sets of each chromosome. So N is going to refer to the number of chromatids. In diploid cells, they have a full set of chromosomes, or 2N. So for example, in humans, we have 23 pairs, meaning that we have 46 chromosomes total, and those pair off. So we have 2 chromosomes on position 1, 2 chromosomes on position 2, so on and so forth. Now, if we're talking about gametes, they're only going to have 1 chromosome on each of those sections. So the gamete is haploid. It has half, diploid, 2. Haploid, half, diploid, 2. So again, the haploid has half the set of chromosomes, and that's going to be represented by our sperm cells. And what is the fancy schmancy biology word for sperm cells? If you said, what is a spermatocyte? You are correct. So, and the proper terminology for our egg cells is called an oocyte. So, some meiosis terminology that I want you to be familiar with. So, you're going to hear me talk about homologous chromosomes. That's going to be our set of 2 of the same chromosomes. We're also going to have a mitotic chromosome. That is going to be 2 sister chromatids that are joined together and present from telophase 1 until they are pulled apart during anaphase 2 in meiosis 2, which we'll get to soon. And then we also have a chromatid. This is a single arm of a chromosome present from anaphase 2 and in each gamete cell produced. Now I'm going to go flashback to some protein synthesis genes and what all of this stuff is wrapped around. So what I want everybody to keep in mind is when we're talking about these different chromosomes and the different structures of them, chromosomes are all of our genetic material. So all these little squigglies right here are condensed strands of DNA that are super tightly wound around these histone proteins. Now, when they get wound around these histone proteins, that's when they actually take on the shape of a chromosome. So just kind of keep that in mind so we know what we're working with. So what exactly is meiosis? It's cell division of the gametes, or of those sex cells. So in meiosis, the nucleus actually goes through 2 different divisions. In meiosis 1, and then again in meiosis 2. This reduces the number of chromosomes in each cell. It also introduces genetic variation through crossing over, and we're going to talk a little bit about that later. It produces 4 genetically different haploid gametes. So because of some of those genetic recombinations, we're going to have 4 totally different gamete cells. And meiosis is not a cycle. Gametes are either used or they're not used. So steps of meiosis. We have interphase, which similarly to mitosis is composed of G1, S for synthesis, and G2. Meiosis 1, we have prophase 1, metaphase 1, anaphase 1, and telophase 1. We also have this kind of weird thing called interkinesis, and that's going to be that portion of time in between meiosis 1 and meiosis 2. Meiosis 2 consists of prophase 2, metaphase 2, anaphase 2, and telophase 2. So interphase is just like the interphase in mitosis. It consists of the G1, the S, and the G2. And as I always tell my students, remember that the S stands for synthesis. Synthesis of what, you might say? Synthesis of DNA. This is identical to the same process of mitosis. So prophase 1 is not completely like mitosis in the prophase portion. So events that do happen in both mitosis and meiosis include that the nuclear membrane does disappear, and then chromosomes condense and become visible. And remember, when those chromosomes are condensing, it's because the DNA is getting super tightly wound around those histone proteins, and that's what makes those chromosomes visible. And remember, those chromosomes are all of the DNA that has come and wound around those histone proteins and come together. So events that only happen in meiosis, we have crossing over that occurs in prophase 1 of meiosis. And in this process, alleles are swapped between homologous chromosomes, leading to some of the genetic diversity. So if you look right here, you're going to see this crossing over process take place. So looking here, we have these homologous chromosomes, and crossing over is taking place on the legs right here. So metaphase, anaphase, and telophase 1. The major events in these steps are pretty much identical to mitosis. Metaphase 1, the homologous chromosomes line up along the midline of the cell. We might also call that the equatorial plate, the equator of the cell, the metaphysio plate. So many different names to confuse you with, but they all mean the same thing. So those chromosomes are lining up in the middle. In anaphase, remember, ana apart, and telophase, we have 2. So metaphase, remember, lining up in the middle, meta middle, ana apart, tela, we got 2. And then we will go through the process of cytokinesis. So in cytokinesis 1, it's at the end of meiosis 1. It is different than mitosis because it results in 2 genetically different diploid cells. So in mitosis, we have 2 genetically identical diploid cells. In meiosis, we have 2 genetically different cells. Now they are still diploid, meaning that those 2 cells still do have 2 sets of DNA or 2 sets of chromosomes. So then we go into that funky thing that I talked about called interkinesis. Interkinesis is similar to interphase, except that the S phase, or where DNA is replicated, it doesn't occur. So the S phase of meiosis, we do not replicate the DNA. So that is the big difference between S phase and meiosis 1 versus meiosis 2. So in meiosis 2, the end result is going to be 4 genetically distinct haploid cells. And remember, haploid means, if you said half, you are correct. So the events in meiosis 2 occur in the same order as meiosis 1. Prophase 2, again, that nuclear envelope disappears. However, in prophase 2, there is not going to be any crisscrossing, no genetic exchanges there through the process of crossing over. In metaphase 2, again, those chromosomes line up on the midline. In anaphase, those sister chromatids pull apart. Again, in telophase, we got 2. And then we have cytokinesis 2. Now in cytokinesis 2, we're actually forming 4 different gamete cells. So if you look at this diagram right here, we're starting off with our human sex cell. Now at the end of meiosis 1, we still have a we still have a diploid number of chromosomes, so full set of chromosomes. Now after meiosis 2, that's when we're going to get our haploid cells. So those all have half of the set of chromosomes. So remember, this is happening in 2 separate cells. So you have the 2 separate cells at the end of meiosis 2, because in meiosis 2, we start off with 1 and we end with 2 diploid cells. At the end of meiosis 2, we end up with 4 haploid cells. This process right here that I also wanted to kind of point out is called spermatogenesis. So if you see right here, you got the little squigglies. That's called the flagella of the sperm tail. And so it results in 4 different sperm. So spermatogenesis, it produces 4 different sperm cells. Now, something you're not going to need to know for freshman bio is that the opposite of that, when we're producing egg cells, is called oogenesis. Now, unlike spermatogenesis that produces 4 fully functional gametes, in oogenesis, we only produce 1 functional egg cell. The other 3 are called polar bodies. and they're basically just kind of the leftovers of the process. We got some cytoplasm in there, some leftover components of cells. Oogenesis, if you kind of made that connection, is a lot more energetically costly than spermatogenesis. So I want you to kind of keep in mind too that females are already born with a finite number of eggs, meaning that if you give birth to a baby girl, that baby girl already has a finite number of eggs that she can use throughout her life. Now, they're obviously not mature yet, but she has just that finite number. Whereas males basically have an infinite amount of sperm because they're continuously producing sperm. Whereas females, specifically of the human variety, they have to wait till those egg cells mature, and we typically go through one egg cell a month. So this is our diagram of meiosis 1 and meiosis 2, keeping in mind as we start meiosis 1 in prophase, this is when crossing over can occur and we're exchanging some of that genetic material. Metaphase, they line up on the middle. We do have those homologous chromosome pairs. So meaning that we have one set still from mom and one set still from dad that have the same genes on them. They might code for different traits, but for example, they might both be carrying genes for eye color or hair color or skin color. At the end of meiosis 1, we are going to have our 2 cells. Those are still diploid cells, meaning they have a full set of chromosomes. A lot of them have been genetically rearranged a little bit to increase our genetic diversity. Then they go through prophase 2, and again during prophase 2, we don't have any crossing over that occurs. Then similarly, we have metaphase, meta middle, ana apart, kilo we have 2. And then at the end of that, we're going to have our 4 gametes. Now, speaking of gametes, this would be an example of our spermatogenesis, producing 4 fully functional sperm cells. Like I said, in oogenesis, we would be producing 1 functional ova or egg cell and 3 polar bodies, which will basically be reabsorbed into the body. So again, why do we want to undergo the process of meiosis instead of just make copies of ourselves? It's important because it does allow for genetic diversity. And it does that by employing several different strategies. Crossing over swaps alleles, and those make the gamete unique. We also have independent assortment that occurs. So what does genetic diversity mean? It means that each offspring has a different combination of alleles. And this ensures that the offspring is different. Unless you're identical twins, then you share the same DNA. What I find interesting about that, though, is that if you do take 2 identical twins and they live in different environments for a long amount of time, so say you graduated from high school and now you're on your way to college, then you graduate college, you live in 2 different cities, one of them might smoke, one of them might not. Their epigenomes are going to be different. And we'll talk more about epigenetics in a different video. So this is good genetic diversity because if we were all identical and a disease came along that could kill all of us, no one would be left to survive. So genetic diversity gives us the ability to adapt, and the ability to adapt gives us the ability to survive. Now, looking at some of the other terms used in meiosis as well as genetics, genes, these are portions of DNA that contain instructions for different traits or characteristics that an organism has. So for example, flower color in plants is controlled by a specific gene, or it can be controlled by a specific set of genes, or polygenic. Alleles in different versions of the same gene. So for example, the flower color gene has an allele that will make the flower purple, but another allele that can make the flower white. So these are the alleles right here that code for the gene of eye color or of flower color in this example. They could code for skin color. So different forms of the allele can create different phenotypes or different presentations of that trait. So traits are the physical manifestations of genes and alleles. And if you remember back to genetics, the phenotype, pheno's like a photo, or at this point, pushing the button on your phone, not the shutter. So that's going to be what the appearance is. What does it look like? So phenotype is like a photo. The genotype are the actual genes. When we're talking about genetics, we typically represent them with different letters like big A, little a. Those are going to be the genotypes, whereas phenotypes like a photo, and that's the physical appearance. So now we're going to talk a little bit about how that genetic diversity occurs. So crossing over occurs during prophase 1 and meiosis 1. So this is when chromosomes condense, and again, when they condense, all the DNA is getting super tightly wound around those histone proteins and forming the structure of a chromosome. So this means that at the end of meiosis, each gamete will have a different set of alleles, leading to genetic diversity. So if you look here, this is an example of crossing over. You see that the crossover is occurring right here, and that's going to lead to genetic diversity. So we can have a recombinant chromosome. This is when alleles have been swapped with a homologous chromosome, and they now have a different genotype than the original parent. In non-recombinant chromosomes, we're not going to have swapped alleles with homologous chromosomes. Those chromosomes have the same genotype as their parents. So these right here have the same genotype as their parents, whereas these have undergone a crossover process. Now, this is going to be coding for the same trait, but they're going to have different alleles. So now instead of maybe carrying the allele for purple flowers, this one might be carrying the allele for white flowers, and this one might be carrying the allele for purple flowers. So we also have a process called independent assortment. So at the end of meiosis 2, each haploid gamete should receive one of each chromosome, 1 through 22, which are our body cells, our autosomal cells, or somal, meaning body cells, and one of each of the sex cells. So mom will contribute an X because that's all a female can contribute, and the male will either contribute an X or a Y. So the law of independent assortment states that alleles located on different chromosomes sort independently of the daughter cells. So for example, meaning that the allele for brown eyes is not required to always be inherited with black hair, or blue eyes always being associated with blonde hair. Now we do have traits or genes that are linked, and we'll talk a little bit about more about that in a different video. So therefore, when referencing independent assortment, traits have an equal opportunity of occurring together, meaning that the chance a gamete will have brown eyes and black hair is the same chance as that organism might have to have brown eyes and blonde hair, so on and so forth. So sometimes things go wrong, and we have different types of chromosomal mutations that can occur. So sometimes meiosis doesn't really work correctly, which can lead to chromosomal mutations. Now, chromosomal mutations are the most severe type because they can lead to a lot of genetic diseases. And this is because entire sections of the genes are deleted or added. And if you remember, when a gene is deleted or added, meaning deletion or insertion, we get a frame shift mutation, meaning that everything changes from that point on, rather than a point mutation, which might just change one single amino acid. So we have duplications, we have inversions, deletions, insertions, and translocations. So in duplication, we have a section of the chromosome that occurs 2 or more times. So it's duplicated. Inversion is when a section of the chromosome is basically flip-flopped. It's reversed. So as you see here, if you look, this lighter yellow band is on the bottom, and then over here, it's going to be on the top. We also have deletion, and that's when an entire section of that chromosome is removed or deleted. Insertion, exactly the opposite. It's when there is a part of a chromosome inserted into a different chromosome. So meaning that it is different than duplication, where this is just a copy of the same section. In insertion, we are actually inserting a different portion of a chromosome into that one. Translocation is when 2 non-homologous chromosomes exchange information through crossing over. Then we also have a process called non-disjunction, and that occurs when the homologous chromosomes, they don't separate correctly. This can occur either during meiosis 1 or it can occur during meiosis 2. And it results in gametes with incorrect numbers of chromosomes, which in turn can lead to genetic disorders such as monosomy, meaning having just one chromosome on each of the numbered pairs, or trisomy, meaning having an extra chromosome when there shouldn't be one. So in non-disjunction, fertilization that involves gametes leads to genetic diseases. Trisomy, like I just discussed, is a condition where an individual has 3 of the same chromosome instead of 2, whereas monosomy is a condition in which an individual has only 1 chromosome. So right here, this is going to depict a normal meiotic division, which results in our gametes. Now, in non-disjunction in meiosis 1, that means that during the metaphase and anaphase portion of meiosis 1, those homologous chromosomes did not separate correctly. So instead of separating out, we have an entire pair of homologous chromosomes here. So when those go through the process of meiosis 2, it's going to result in gametes that have an abnormal number of chromosomes. So here we have 3, and here we only have 1. Now in non-disjunction that occurs in meiosis 2, during meiosis 1, business as usual, everything separates out, we're good to go. But all of a sudden, in meiosis 2, during anaphase 2, same thing happens, goes to split apart, and then 2 of them remained intact. Could be more than 2. And again, that's going to result in abnormal gametes. Now, it's only going to result in 2 being flawed versus all 4 when there's a non-disjunction that occurs in meiosis 1. So, I hope that was helpful for everybody. If you guys want to see any weird, dorky videos from the Queen Nerdling starring some of her nerdlings themselves, you can follow me on TikTok or Instagram. I will see you soon. Stay nerdy till next time.